Good day everyone. Welcome to the week 5 of the course Biological Data Analysis and Visualization with R. In this week we will be taking on another problem which will be chip sequencing data analysis with R. Okay, so that is a new topic that will be first introducing in this class. What we will do is we will first talk a little bit about chip sequencing and what is chip sequencing, why do you do chip sequencing. So, that is the topic for this class, and then we will take again some hands on problems, right, which we will solve through the through this week. Okay, so, in this class I will very briefly introduce the ChIP-seq method, what ChIP-seq method is, and then I will be talking about the data analysis step. Okay, so, some of you might be familiar with ChIP-seq experiment. What is a ChIP-seq experiment? But just to be on the same level for everyone, I will describe very briefly what is ChIP-seq experiment, what are the objective of ChIP-seq experiments. Okay, so, let us begin with that, and then we will talk about the data analysis steps very briefly, and then we will then start with the hands on analysis. Okay, so, first what is the full form of ChIP-seq? So, the ChIP stands for chromatin immunoprecipitation, and then we have seq part, that is the combined with sequencing. And so, this sequencing is usually next generation sequencing. So, in many of the platforms that are available, but most commonly it is in the Illumina platform. So, the chromatin immunoprecipitation, right. So, we have some sort of antibody against protein or some modifications that allows us to identify these protein binding sites in the DNA. Okay, so, that is the idea behind this experiment very briefly. So, that is why the term immunoprecipitation, because we have an antibody involved. Okay, so, what are the objectives of a ChIP-seq experiment? So, very briefly we do ChIP-seq experiment if you want to identify genome-wide identification of the location of DNA binding proteins, right. So, these DNA binding proteins could be, for example, transcription factors, right. So, one of the questions that we are really interested in often is where are the transcription factors binding in the genome and how do they affect gene expression. Okay. So, we have a lot of gene expression data, but then we want to also know how that expression is determined by the binding of transcription factors. So, for doing that we do this kind of ChIP-seq experiment, and we actually want to do this in an unbiased manner for the whole genome, and in that case we will do a ChIP-seq experiment. So, these DNA binding proteins would also include chromatin remodelers, right. So, these are again important for gene expression, etc. So, this study, right, ChIP-seq, the binding sites of transcription factors and chromatin remodelers, this is part of epigenetics. We also are interested in location of specific histone modifications. Okay, so, in epigenetics there is a lot of interest in knowing what kind of modifications are present in different regions, for example, in the promoter of a gene or in the gene body itself, right. So, what kind of modifications are there, right, because these modifications can impact transcription factor binding, expression patterns, etc. right. So, again this is something that is a very important part of epigenetics. Okay, so, some of the histone modifications, just to give you some examples, like H3K9 AME3 or H3K9AC, etc. right. So, you have many such modifications that can be studied, right. So, the goal of these experiments is like to identify. So, with ChIP-seq we can actually identify all of these location of these modifications or location of these transcription factors or chromatin remodelers in the genome, right. So, this is something that is very important for epigenetic studies, and then of course, we can then use this information to connect location of these modifications, right, with gene expression patterns, right, and this is something that is interest to many scientists. So, again just a very brief background, as I said, ChIP-seq, right, chromatin immunoprecipitation with sequencing, but then there is an alternative idea which is the chip on chip, which is called chromatin immunoprecipitation with DNA microarray. Before next generation sequencing, researchers used to do these experiments with DNA microarray. But we will not go into all these microarray experiments or next generation sequencing, because this is not the topic of this course. But if you are interested about next generation sequencing specifically, we have covered that in a different course. So, let us now very briefly look into the experimental design, because this is something that is very important when you do the data analysis, right. So, that we understand how the data is generated and what kind of controls are there, so that we can properly do the data analysis. Okay. So, how does chip sequencing work, right? This is kind of the idea. So, you can imagine this is the chromosome or the DNA, right, where you have this histone bound, right, histone proteins, and you also have, let us say, transcription factors or chromatin remodelers that are all present, right. So, these are different DNA binding proteins, and then you have these modifications on histone, right. So, you have a methyl modification, you have acyl, right. So, AC modification, ME modification, right, all those modifications are there. So, what happens is, first we let us say fragment the DNA, and when we fragment the DNA, we get these different fragments, right, and there are some bound fragments, right, DNA that are bound by, let us say, transcription factors or DNA binding proteins, and some of these fragments are bound by histones with some modification. Okay. So, this is how the protocol works, right. So, when we are doing immunoprecipitation, means we want to identify a specific class of protein, right. So, it's either could be the transcription factor, for example, the blue transcription factor, blue protein here, or this golden protein here, or maybe this blue histone with this methylation mark. Okay. So, we are interested in location of any of these proteins or these modifications in the genome. Okay. And based on that, we design the antibody. Okay. And that is where the immunoprecipitation part comes in. So, let us say if you are interested in this ME modification or methyl modification, right, histone ME modification, we will design antibodies that will bind this specific modification. Okay. On the other hand, if you are interested in, let us say, in this greenish yellow DNA binding protein, then we will design antibody against that protein, right. So, again the specificity of this experiment is determined by the antibody that we have, right. So, that can be a good thing or a bad thing, right. If you have a very specific antibody, of course, you will identify these specific binding sites more accurately, but if you do not have that specific antibody, you might identify a lot of extra sites as well in the analysis, right. So, these are something that we need to also keep in mind. Now, once we have identified this antibody, so we treat them with antibody, right. So, we have this affinity binding, right. So, we have antibody bound fragments which we can now separate out, right, and rest of the things are washed away, right. These are not bound by antibody, so they would be simply washed away, and then we take these fragments that are bound by antibody, and then we purify them, and then we sequence them. Okay. So, I think the first part is clear, right. So, we now understand the immunoprecipitation part, right, the chromatin immunoprecipitation. Okay. So, here what we are doing is we are taking a specific antibody, and this antibody is designed so that we can identify our proteins of interest or our DNA modification of interest, right. So, H3, let us say H3K9Me3 modification that we are interested in. So, we will design antibody against that H3K9Me3, right. So, based on this antibody, we have specific target in our mind, and we do this experiment where we separate out DNA fragments, right, genome fragments that are bound by this protein or have this histone modification, right. Once we take them out, right, we then go for next generation sequencing. Okay. So, when we do the sequencing, now what we are going to do is we are going to describe the next steps. Okay. We have done the sequencing, what is the challenge now. Okay. So, let us say we take both these examples and we will kind of illustrate what is the challenge here. So, here is the, let us say, first idea, right. Here we have done the sequencing from both ends, right. This is the paired end sequencing in any platform that we like, and once we do the sequencing, what we will end up with are bunch of reads here. Okay. So, this is something that is important for us to understand, right, when you go into the hands on, etc. what's the major challenge here, okay, and what kind of output we will get and what kind of analysis we will have to do. So, here the bunch of reads, right, these are the reads are simply DNA fragments that are generated by sequencers. Okay. So, these sequencers will give us the most common sequencer like Illumina, it will give us short read fragments, right. So, between 100 to 250 at most fragments, right, size would be 100 to 250 base pairs, right. So, these are the reads here, okay. These are the DNA fragments that we get from the sequencer, and similarly here we have the sequencer. Now, the thing is, right, we get these fragments from the end of the DNA. Now, the question is, where is the exact location, right? Based on these reads, we cannot really determine the exact location or the binding site of this DNA binding protein, for example, or the location of this histone modification. Okay. What you can say, okay, this is located somewhere here in between these reads. Okay. Now, you might ask, right, this might vary depending on where from we are sequencing, right, and of course, this is dependent on if we are sequencing from close to this site or far away from this site, but we usually do not have any control over this site in simple ChIP-seq experiment. There are experiments which can tell us, right, that can give us that choice where we sequence very close to the binding site. In that case, this problem is resolved. Okay, so because we are focusing on ChIP-seq, right, so this is one of the major issues that we have to deal with when we do the data analysis. Okay, what we know is that, okay, this modification is somewhere in between, right. This is something we know, and here we also know, okay, this is somewhere in between, the binding site is somewhere there. The question that we are interested in is what is the exact location of this binding site or the histone modification. Okay. To do this analysis, right, we have to do something called peak calling. Okay. So, this is an important step in ChIP-seq data analysis, and we will see when we go into the hands-on part, we will do the peak calling part. Okay. There are tools that will help us do this peak calling. Okay. And that is a very important part of the computational analysis. Okay. So, what does peak calling do, right? From this data, right, from this read data, it will predict, right, where the peak is, where the binding site is, right. So, this is something like an estimation or kind of like predicting the binding site based on the distribution of the reads in the genome. Okay. So, this peak calling part is a very important part, and this is determining the binding site or the site of histone modification. Now, the resolution again depends on the, as I said, on the fragmentation. Okay. So, where you are fragmenting, right, and from which site you are sequencing, right. This is a very important part, and this resolution depends on the fragmentation size, but it is usually between 50 to 100 base pairs, right. So, we get maybe we can determine within 50 to 100 base pairs, this is where the binding site is. So, we will see again in hands-on, right, we will have some sort of uncertainty, but the actual binding site, this transcription factor binding sites or the modification sites, these are usually only 10 to 12 base pairs. Okay. So, especially for the DNA binding protein. So, how do you actually go into the 10 to 12 binding, 10 to 12 base pairs resolution compared to 50 to 100? Okay. So, this is where peak calling kind of tells us, right, kind of predicts, okay, can help us, right, pinpoint the binding site. There are other ways to do this as well, which we will not discuss in this class. Okay, so we now understand, right, what we have to do. We now understand the experimental part, we now understand the computational part, and what are the challenges that we have in this data analysis process. There are also some more experimental details that we should know. So, for example, we need to do control experiments for ChIP-seq. So, there are 3 types of controls that could be there. Usually at least one type of control would be there in the data, and we will see, like we will analyze data, we will have one type of control. So, the control that we definitely need is the input DNA control. Okay. So, that as I said, the ChIP-seq depends on the reads binding to specific reads coming from specific region of the genome, right, where we have the DNA binding protein present or we have the modification present. So, this analysis is dependent on peak calling, which is dependent on enrichment of reads from specific genomic regions. Now, how do you calculate this enrichment? This enrichment is calculated compared to the input DNA control, right. So, this enrichment can come from even from random sampling effect. When you do next generation sequencing experiments, we have this variation in read coverage or read depth across the genome, and to distinguish these random fluctuations from an actual peak, we need to do an enrichment analysis. So, we do the enrichment analysis from the sample, right, our treatment or the sample versus an input control. Okay. So, all experiments, all experimental data that we will be looking at, they will have a control, okay, and usually that input DNA control. So, this input DNA control, this is prior to immunoprecipitation. No immunoprecipitation is done, it is just before the immunoprecipitation, that is the genomic DNA or the input DNA. In some of these experiments, you will also see that there will be other types of controls, for example, DNA from mock immunoprecipitation without any antibody. Okay. And again, this is what we are, why are we doing this experiment, or why would anyone do these experiments, is to look for enrichment just because of the immunoprecipitation process, right. So, again, immunoprecipitation process will select only a certain number of reads, right, and that might induce some sort of enrichment, but that is not a true enrichment because no affinity purification or immunoprecipitation has been done without. So, there is no antibody. So, of course, this enrichment is just because of the sampling effect. So, all these controls try to understand, right, the enrichment from the process, the sample preparation process itself, okay, and the goal is to kind of distinguish this technical variation that we generate from actual biological variation. Okay, so, the third type of control that can be there is DNA from non-specific immunoprecipitation, right, where we can use a non-specific antibody that should not enrich for these binding regions of this protein, right. Again, this again is accounting for the non-specific enrichment. Okay. So, all these controls, as you see, they are kind of trying to looking at the technical variation or the enrichment due to the sample processing steps. In most cases, right, in many cases you will not see this control 2 or control 3, but control 1 would always be there, right. Without that, you cannot do the enrichment analysis. Anyway, so this is something that has been discussed in much more detail in one of our earlier courses. Now, once we understand the basic principles of ChIP-seq, what we are going to do is we are going to discuss the data analysis workflow. Okay. So, how do you process this ChIP-seq data? Okay. So, the first step is, of course, as I said, we do the experiment, we do next generation sequencing, right, after the immunoprecipitation step, and we sequence using a next generation sequencing platform, we get the read data. Okay. The first step that we have to do once we get the read data is data quality control or QC, and this is this can be done in two different ways, right. So, one is quality evaluation and this is data pre-processing. Right. So, there is an elaborate data quality controls process for all the data that we get. The idea behind all this data quality control is to make sure that the data that we are working with is of good quality. Otherwise, whatever inferences that we are going to draw at the end of the analysis could be erroneous, right. So, that is something that we want to ensure before jumping onto the actual analysis, that okay, the data that we are working with is of good standard. And to do that, we use two different metrics, right, different types of metrics, right, and they can be classified into broadly two classes, right. One is the quality evaluation and the second is data pre-processing. So, the quality evaluation is looking at the actual quality of the data, whether there is any contamination, whether there is any bias, right, when we are sequencing the DNA fragments. So, that is the quality evaluation part. The second part is the data pre-processing part, right, where if we see some sort of contamination, for example, let us say adapter contamination or some sort of quality issues, we can get rid of those, or we can do some pre-processing so that we can address those issues effectively. Okay. So, this is something that is very important, but we are not going to discuss all this in this class. Once we have done this, is the next step is we do a read mapping against the reference genome. Okay. So, if we are working with human genome, then we need to map the reads against human reference genome, but if we are working with other organisms like mouse or Drosophila or plant, we need that reference genome. Okay. So, we take the reads and map against the reference genome. What we get is usually a SAM file, and whose binary version is the BAM file. Okay. And this is the first few steps, right. So, this is a very important step, right, the alignment, and the BAM file, this is where we will start the chip sequencing analysis. Okay. The first 3 steps we have discussed in an earlier course, and this is something which we are not going to do in this class. Okay. So, once we have this BAM file, we can generate something called BWIG or BW file. This is something where we will know where the reads are mapping in the genome, and this is something that helps us visualize the data. Okay. From the BAM file, we can do binning and peak calling. Okay. And hopefully you remember, right, as I discussed, this is a very important step. So, binning is simply looking at different windows of the genome, right. So, if we have like a large genome, we fragment it into different beans, right, different sizes with specific size, and we kind of count how many reads are mapping into each bean. Okay. So, that is the binning part, and from this information we then do the peak calling, right. We identify the peaks of the reads, okay, where the reads are enriched. Okay. So, we will see this in hands on, and we will get a better idea of the whole process. Once we have done this, right, what we can do is we can visualize these peaks, right, and we can also visualize the BW file. Okay. And once we have this peak calling, we can then do something called a differential binding analysis. So, what is differential binding analysis? It is something like let us say we are working on 2 different sample types, and we are interested in looking at the binding pattern of a transcription factor in these 2 different sample types. Okay. And we will do ChIP-seq experiment on both sample types, and then we will identify the peaks, and once we have identified the peaks, we can then kind of ask, like, whether the peaks are similar or whether there is any change in the location of the peaks in the genome. Okay. And this is what I mean by differential binding analysis, right. Similarly, you can ask, right, whether the histone modification patterns have changed because of some treatment, let us say, or maybe because of different sample types. If you take the earlier example, right, cancer versus normal, whether there is any difference in the pattern of these histone modifications. Okay. So, all those things will come under the differential binding analysis. Okay. So, what I am going to do is I will just give you an overview what we are going to discuss in this course, right, and in the subsequent classes. Right. So, as I said, right, we have talked about the first 3 steps in a lot more detail in one of our earlier courses on next generation sequencing, and how to what are the different types of data that we get, right. For example, single end sequencing, paired end sequencing, all those things, these are discussed, and different formats for the data, right, depending on the sequencing platform, whether it is Illumina, let us say, or we was working on PacBio, SMRT sequencing, etc. So, this is something we have discussed in a lot more detail. We have talked about data quality control, and how to visualize these quality control values, right, the different parameters that we assess, and we have talked about the data pre-processing part, right, whether we have to do trimming, etc. All those things have been discussed in the earlier course. We have also talked about different tools that we can use to do the read mapping. Okay. So, this is again we have discussed and how to go to a SAM file or a BAM file. So, we have discussed this in earlier course. So, if you are interested, please feel free to take a look and try to understand. What we are going to cover in this course are these 2 very important parts. Okay. So, the first part is the binning part and the peak calling part, right. So, we will start with BAM file, and we will do the binning and the peak calling. Okay. And once we have done this, we will then do the visualization to actually see where the peaks are in the genome. Okay. So, we will take a data set for doing precisely this, right. So, we will have the BAM file, and we'll go through the binning process, peak calling process, and visualization. One thing we need to remember, right, when you do the peak calling part, we'll have to have a control, right. So, peak calling will be done compared to a control sample. Okay. So, that control will be there, and it is usually the input DNA control, as I mentioned. Okay. So, and then we will look into the genomic tracks, we will identify, we will see these peaks, location of these peaks and where they are located, what are the genes that are present in those locations. So, we will take some examples and I will illustrate that in the hands on part. The other part that we will also do is we will take a BW file, right, big file that is often deposited and generated from BAM file, because these are much more compressed files, and we will take a BW file and we will visualize the data, because for many cases we will see these BW files associated with data sets that are submitted, and we want to see the pattern, right, of this read map, the mapping of the reads, etc. Okay. So, this is something that's what we are going to do using two different data sets. Okay. So, in one of the hands on, we will first start with BAM file, right. This is something that will be very useful for you, right. If you are doing your own ChIP-seq experiment, you will get this data, then you can do the data quality control, you can generate these BAM files and how to proceed, right. So, the first steps we have discussed in earlier course, so you can follow that and arrive at this step. From this BAM file, how can you do the binning, peak calling and the visualization? This is something that we are going to cover here in this course. So, in one hands on will be focused on this part. The other hands on will be taking this BW file, right. So, these BW files are generated from these BAM files, and so BW files, and then we will visualize them. So, we will also talk very briefly about this big wave format, what this format means and how we can visualize this data from this format. Okay. So, that's the discussion about ChIP-seq. So, from the next week, next class we will be talking about the hands on part, we will be talking about the data sets, the packages that are there, and we will be completing this part. So, here are some of the references that we have mentioned here, for example. So, for the first few references, right, you can look at the ChIP-seq experiments, right, and what are the advantages, challenges, etc. And then in the next part, right, we have some workflow, reference to an workflow, for example, here a practical workflow for ChIP-seq analysis. So, this is something that would be useful to understand the data processing pipeline. So, to summarize, we have talked about ChIP-seq experiments, and ChIP-seq allows us to do genome-wide mapping of DNA binding proteins and histone modifications. So, this is a very powerful technique in epigenetic studies, and the protein of interest, right, whether we are working on DNA binding proteins or histone modifications, right, this is dependent on the antibody that we are using. We have talked about the experimental pipeline as well as the data processing pipeline, both are very important, right. In the experimental pipeline, we need the input data. In the data processing pipeline, we process the next generation sequencing data that is generated after the experimental step, right. Again, this data processing pipeline is also critical, and there are several points that we will be discussing in the hands on part, and this is something that we do to actually identify the peaks of binding. The final goal is to identify these peaks, right, where the DNA binding proteins are bound or the histone modifications have taken place. And we will also see that we will also do some visualization, because this is very powerful to actually give us some insights into the changes in DNA binding or the histone modification patterns that is happening across different samples or across treatments, and we can see them visually, and we can then of course correlate or corroborate with the peaks that are identified by the software tools. Thank you.